next speaker is Shian Su. Shian Su is a PhD student in bioinformatics at Moderator Lab, WEHI. Shian is the maintainer or one of the authors of multiple bioconductor packages, for example, NanoMesBits or CellBench or Glimma. Especially for Glimma, an article about the new version is published on the preprint server. So I'd like to turn things over to Shian.
Thank you, Kozo, and thanks to the Bioconductor Asia Committee for allowing me to give this talk. So today I'll be speaking to you about Glimma version 2, which is an update to our Glimma package that's been in Bioconductor for more than 5 years now. And the main feature of Glimma version 2 is to introduce Markdown integration to the existing plots that we have. And also it includes quite a few major updates in order to make that possible. Oop, how did I change? Sorry, I'm having trouble switching slide. There we go. So Glimma creates plots for the purpose of RNA-seq analysis. And to start from the beginning, this is the central dogma of molecular biology, where we have our DNA, and that is transcribed into copies of mRNA as we need. And those mRNA serve as instructions that get translated into proteins, which perform most of the tasks in our body. And by extension is an important factor in all sorts of diseases or other health issues. So mRNA, we've gotten quite good at sequencing and quantifying those molecules. And so from RNA sequencing, we're able to take all these messenger RNA molecules and quantify the level of expression of particular genes in our body. And from these aligned sequences, we can, from the sequences, we can align them to our genome and find out which genes are being expressed. And from that, we get a big table of these gene counts. And as bioinformaticians, we like big tables of numbers because that's usually the starting point of a lot of statistical and other types of analyses. So in this example, our gene counts has information about a lot of individual samples and a lot of different genes and the number of transcripts that we have discovered in our samples. Then differential expression is one of the basic type of analyses we do in RNA-seq. And what that involves is asking the question, how does gene expression differ between different groups of samples? So in a common situation, we may have samples 1, 2, 3 be a part of a control group and samples 4, 5, 6 as a part of a disease group or treatment group or other type of group. And we would like to know which genes have different expression levels between those two groups. And to do this, we can use a variety of methods available on Bioconductor. So if we have a gene count matrix, then we can use Limma, Edger, or the seq. And those will tell us among the tens of thousands of genes that we have profiled using RNA-seq, which of those are differentially expressed between our samples or between our groups of samples. So what that usually results in is a plot that looks like this on the right, where we have for each dot represents a gene, and we're plotting these mean expression across our samples against the log fold change between our groups of samples. And what that will tell us is, a certain number of genes will be upregulated in the comparison, and a certain number of genes will be downregulated. So when we perform this analysis, the result that we generally present in a static form looks like this. We'll email something like this off to a collaborator who did the biological experiment, and they'll see a plot that shows roughly the number of genes that have been upregulated and downregulated. And then they'll have this big table of results with more detailed statistics. Might be a bit hard to see, but each row is a gene. It's got some descriptions of the gene, what chromosome it comes from, log fold changes, p-values, and other important information. And so once they receive this, what we find is they often have very much the same question upon seeing this data. And they come back to us and they ask, well, what's this gene over here? What's that gene over there? And where's my favorite gene? And we will have to go back and figure that out. And there's a lot back and forth. They may, after asking this once, they might ask it again for a different gene. And so there's a lot of back and forth, a lot of extra work required for the collaborators to finally figure out the things that they want. So the motivation for the original version of Glimma was to produce something that was interactive and allow the collaborators to explore their own data and link up the information between the plot and the table, such that these questions can be answered by the collaborators themselves without having to come back to us. And these are just some animations from the first version of Glimma showing the things that we do. So there's two main plots that we produce: an MDS plot, which is a multi-dimensional scaling plot, very much similar to a PCA plot and these days TSNE plots, where we have each sample as a point, and there's going to be separation based on the gene expression profile of those samples. So in the original version of Glimma, we provide the ability to change the dimensions you're plotting. Very commonly, you just plot the first two dimensions, but you never explore the higher dimensions for other information. And we also provide the ability to change the grouping variable that you're coloring the points by. And that just allows you to do a more in-depth investigation of the factors affecting the clustering of your data. So in this case, we just have the biological group and the batch. And often batches will reveal some sort of major separation between your data. The other plot that we provide, and the main one of the Glimma package, is the, at the time, MA plot. These days it's MD plot. And what it is what I showed before. It's the plot of the average expression of the genes against their log fold change between two comparison groups. And from this plot, you can mouse over the individual points, and that will bring up a tooltip. And it will show you information about that point, as well as highlight that corresponding gene in the table below. So from this table, you can also actually click on the individual rows, and there's a search bar, and you can search for your favorite gene, click it, and it will show up on the plot above. So that's the linking between the information. It's a two-way linking. As well as that, you can also see on the right that we will plot the expression levels of the particular gene within each sample, separated by their group. And that's also a very nice other point of information that you can make use of. And on top of all that, one of the other primary goals of this was to make all of this portable. So at the time, we didn't want to use Shiny because you can't really provide that to a collaborator who doesn't use R and have them be able to actually explore that data. So all of this has been produced as just a standalone HTML page produced out of R that you can put into a folder and send off to a collaborator. And so the original implementation of this used the D3 library from JavaScript. And we took the data from R and we exported it as a JSON object, which then we injected into this template JavaScript HTML page that used D3 for the plotting. And unfortunately, at the time, there wasn't really that much better available. These days, there's many more libraries, which motivated the rewrite that I'll show you. But the main limitation was that D3 was very low level. It wasn't really a plotting library so much as it's a graphical library. You can do very flexible things with it. It's very low level. You're just drawing circles and lines, but it doesn't just create a scatter plot. It doesn't create a bar plot for you. So that made producing new features, debugging, and just various changes a lot more difficult, and eventually became quite unmaintainable as I moved on to other projects. So Glimma V2 is a major rewrite that has been basically tore up the whole thing and just rewrote it in a different library, where we've replaced the custom HTML and D3 with HTML widgets and Vega. So Vega is once again a JavaScript library, which actually is based on D3, but provides much higher level functionality. And this allows us to actually implement features much easier, makes things easier to maintain, and just gives us more features overall. So now in the new version, the MDS plot looks like this. So things to take note of is that we have a more flexible selection of dimensions we can plot against each other. Previously, the only option was to plot two adjacent dimensions against each other. So, dim 1 versus 2, 2 versus 3, 3 versus 4. But now you can do 1 versus 3, 1 versus 5, and whatever you want to choose out of the dropdown. We've got different scaling, so different symbols, different scalings, and just a lot more options thanks to the use of the new Vega library over D3. On the MA/MD plot side, we also have new features, a legend that's been very widely requested for quite a while now that that I never figured out how to implement back in D3 in a way that I was satisfied with. It's now just an included feature of Vega, so that's just there for free, which is great. And now we can also select multiple genes. And what it does is as you click through the genes in the table, it will annotate them on the MA plot. And also for the gene that you most recently selected, it will draw the expression across the samples as it did before. So with the new Vega functionality and HTML widgets, all of this now integrates very well with RStudio. So previously, this would pop out a web page in whatever default web browser you had. Now it just pops out in the RStudio viewer, which I guess one less program required to view these. And it all works as I showed before. And more importantly, it also integrates with Rmarkdown. So previously, the plots in Glimma version 1 would have to live on its own web page, so it's its own HTML file. But now it can be integrated into a Rmarkdown report, which is, I think, gold standard these days for reproducible R analysis reports. So if you were to use these Glimma functions, these Glimma version 2 functions, so this is actually just a screenshot out of a Rmarkdown report. So, above you've got your Markdown with the text with your code, the code that's run, and then immediately after you have the result of that code, which is fully interactive within the report. So now all of the Glimma plots work inside of our Markdown reports. You can compile the report and you can send it off to people, and all the plots you create are fully interactive with all of their features available as part of the report. And if you would like to actually save the Rmarkdown reports, if you wanted to save those plots as their own individual HTML pages as you did before, that's still possible. So if you take one of our plots, you save it as a variable, you can actually use the HTML widget save widget into a HTML page, and that will allow you to save those individual plots into their own HTML pages as you would in Glimma version 1. In addition to the new integration with HTML widgets, so usability with RStudio Viewer as well as Markdown, we also, through the Vega library, are able to provide some data export functionality. So if we look at the MA plot, a lot of the time in Glimma version 1, I think what we would see was that people would just take screenshots of the plot created in Glimma, and then that would make its way into a publication. And they look fine, but I always cringe a little when I see people put screenshots out into publications. So now we allow them to actually export. So down here, there's a save plot function, and that's provided through the Vega library. So we actually allow you to export either of the plots that you create or that you've annotated or otherwise queried. And we allow you to save either of these plots as a PNG or an SVG. And the SVG will allow you to create much higher resolution, higher quality plots that you might want to use for publication, because sometimes PNGs get a bit blurry. In addition to that, we also allow you to so you can select a bunch of genes, and then using the save data button over here, we allow you to export that particular part of the table as a CSV. So we can see the option save with a 4. So that's just saving the information for 4 genes, or you can save all. So you can take the entire table and export as CSV to explore in other ways. And so here's a summary of how Glimma version 2 looks at the moment. So we've taken all the previous functions, we've updated them, and we still maintain all the support for the different, I don't know how clear it is to see for those watching this on a laptop. But we can take counts matrices, we can take DG list, edgeR objects, we can take DSeq datasets and the various lemma and edgeR. So we've got MRA, LM, DGE, LRT, and DG exact. So these are the analysis output objects. So we have full support for all of those. And you can plug it into the Glimma NDS plot or the Glimma XY, MA, or volcano plot functions. And all of those will have all of the functions I just showed you and give you the ability to embed them into our Markdown page, as well as export the individual plots as SVGs and PNGs. And yes, Glimma Volcano is something that's also been requested for quite a while now that I never got around to importing. So that never got around to implementing, but now that's available. So hopefully someone's happy about that. And so with the functions updated in version 2, we've actually kept all of the functions from version 1 still in the package. So anyone who's using Glimma at the moment, or they were using Glimma version 1, don't have to change anything in their code. All of the original Glimma plotting functions are still available for backwards compatibility, but I've kind of removed them from the documentation and they will no longer be updated. I'll fix major bugs if they come up, but for all intents and purposes, they are being deprecated, but I will keep them still in the package just for those who have old reports that they might want to rerun. And now, if you want to use Glimma these days, you should be looking at these new functions. So rather than the GL plot series, you want to use these Glimma prefixed plot functions. So Glimma MD and MA, same function, different names, MDS, XY, and Volcano. And in general, they take the same arguments. Some of them require fewer arguments because we saw the opportunity to cut down on the number of arguments you need, because a lot of the data is actually already available through the other objects that we were previously taking. If you want to learn more about them, please go see the vignettes. There's quite detailed documentation there about how these work. And in terms of availability, it's been available since the previous release of Bioconductor in version 2. We've made some more changes in the current version. So if you want the latest and greatest, go to Bioconductor 3.14 to get the latest. I think it's in Glimma 2.4 now. And there's a developed version with example GIFs that we have on the GitHub page for this package. And we've also recently put up a preprint at a archive titled Dashboard Style Interactive Plots for RNA-seq Analysis, Rmarkdown-ready with Glimma 2.0. So please check that out if you get a chance. So with that, I would like to thank Haas and Oliver, who did most of the work in the new version of the package. I basically provided just an advisory role. Charity Law led the project to get all of this done, to update and modernize the Glimma package. And obviously Matt Ritchie, our lab head, provided the support and the initial push for this update. And I obviously would like to thank all of the Ritchie lab, so my lab, for providing a lot of feedback, as we talked about this in our lab, members of the Bullet Lab also at WeHi and the Bioinformatics Support Unit at WeHi who used our package and also provided a lot of feedback. And obviously, anyone who submitted a GitHub issue with bugs and feedback and suggestions for what we should do with our package. So thank you very much. Questions?
Thank you, Shian. If you have any questions for Shian, please write it to Slack. There is a question from Kokitsuzaki. Do you have any plan to use other dimension reduction methods other than MDS?
Yes, so that's something that's in the plans. We are trying to improve support for more single cell applications. So there is actually a vignette on Bioconductor describing how to use this for single cell. There's some issues with the volume of data because we keep a lot of data in the HTML page and that gets quite big for single cells. But we do have plans for TSNE, UMAP, PCA, which we don't think should be too difficult to implement.
Thanks. There is another question from Jeremy Silva. What are the reasons behind plotting the results in Vega instead of Plotly or RBokeh or HighCharter?
So I think we played around with a few of those, and it comes down to, I guess, the simplicity of the libraries. So Plotly provides a bit too many features. It's a bit too feature rich. There's too much going on. It doesn't quite fit the style that we wanted. And yes, obviously RBokeh, and there's many other libraries for this HighChart as well. Some of them I think didn't provide the interaction. They didn't expose the interaction APIs that we wanted. And I think when we just looked at quite a few of them, we found that Vega was just the best fit for what we wanted.
Okay. Any other questions? So I have a question. Can Glimma be used only for the output of Glimma and HR and RSeq, or can be used for more than those?
Yeah, so by default, I think the MAMD plots make the most sense for those packages, but the Glimma XY plot basically lets you plot whatever you want on the XY axis and provide whatever kind of expression matrix you want. So you can make your own custom plot. And if it's something that, oh, sorry, my mic fell out. Can you still hear me?
Yes.
Okay. Sorry, my mic fell out. Yeah, so you can make your own custom plots with things from other packages. And if you think it's popular enough, put a GitHub issue and ask us to support it for any other package that you might want to use.
Okay, thank you. Is there any other questions for Shian? There is a new question from Kokitsuzaki. How many cell numbers can be supported when applying Glimma to single cell data?
Yeah, I think so. When I was playing around with this on a 2016 MacBook Pro 15-inch, it starts struggling quite heavily around 500 cells. And so if you look at the vignette I've put on Bioconductor, I suggest some pseudo bulking, which reduces a lot of these cells down into pseudo bulk samples, which makes it a lot more tractable for Glimma.
Okay, thank you. It seems we ran out of the time, so I would like to finish this session.
Thank you.